Dear students, we have started already to study the mitochondria in the course of our cell and molecular biology. Now we will continue the lecture number 10. Actually, we have started previously the same about mitochondria. It is one of the different cell organelles. It has outer membrane, inner membrane, then matrix in which the cristae are there, or we can say the working unit in the mitochondria. In that we have seen the structure with inner and outer membrane, then perimitochondrial space distance about 60 to 80 degrees angstrom, and these are the lamellae or cristae, which are the basic unit which are working as a metabolic activities, then respiratory activities, then energy production, ATP production, everything has been there. We can say the main activities of the cell are regulated by these mitochondrial matrix and mitochondrial organelles. These particles which are attached on the mitochondrial cristae are F1 particles generally, or we can call it oxysomes. Then DNA molecules, circular, they have 7 days ribosomes. We have already studied this in previous lecture. Then we have also seen the structure. Here the cristae, we can see the ribosomes, or we can say mitochondrial matrix having these F1 granules. So here these are being known as oxysomes. So this is the basic structure. So this basic structure of the granules, which is arranged around this cristae, is as such because one particle have a globular head, then a pedicel and base. They are attached to the upper membrane, upper side of the inner membrane, and likewise this is the basic structure of mitochondria, in which the microscopic structure, then internal structure, and then crista, and this one is F1 particle. So this is all about mitochondria that we have already seen. We have also studied the functions of mitochondria in previous lecture. These are respiratory organs. These are the actual respiratory organs of the cell. Then they can metabolize by oxidation the foodstuffs. Then they also create some energy which is released in the form of ATP. Then it acts as a site of oxidation, phosphorylation. Mitochondria also provides a site of Krebs cycle. Then it provides site for dehydration. It provides site for glycolysis. It acts as classical example of the cytoplasmic inheritance, and occasionally it functions as a store organ. Example, mitochondria of ovum stores yolk protein and ultimately gets converted into yolk bodies of egg. So this, up to this we have studied. Now next is the objective questions which you have to solve as per the syllabus you have already studied or lectures we have listened. Then ER acts as secretory storage, or we can say circulatory dash system of the cell. So it generally, endoplasmic reticulum acts as a nervous system of the cell because it regulates the main activities. And likewise dash is called autonomous unit of life, that is plasma membrane is autonomous unit of. Likewise this you have to study. These highlighted are the answers. Highlighted are the answers. So you have to read it as per the examination. We will take a test in the next week of 10 marks. Objective test will be there. So you prepare yourself. In 10th lecture, lecture number 10, we will continue now unit number 2. That is cell division and cell signaling. So as usual, we are well known about the cell division. This is a process of dividing the cell and preparing, or one cell gives two divided cells either in n number of chromosomes or 2n number of chromosomes. And these divisions are of two types, that is mitosis and meiosis. This division is according to the types of cells, because mitosis occurs in body cell or somatic cells, and meiosis occurs in germinative or reproductive cells, and therefore the two different names are given. So what is cell? What are the activities of cell? What is the total life of cell? What is the cell cycle? How cell regulates its all activities? This has to be studied in this chapter, and prior to that we will see what the cell division is. Cell division is the process of cell go through to divide. There are several types of cell divisions depending upon what type of organism is dividing, because every organism have different types of cells, and these cells have different structures, shape, life, everything, the number of chromosomes. There are so many differences. Organisms have evolved over time to have different and more complex forms of cell division, and most karyotypes or bacteria use binary fission to divide the cell. Means they divide directly, vertically or horizontally, one cell into two cells. Then eukaryotes of all size uses eukaryotic cells of all size uses to divide itself by a process of mitosis, and the sexually reproducing cells, or the cells which have sexually reproduction in their germinative organs, are usually of the method called as meiosis, and this division is to reduce the genetic content in the cell, or we can call it as a reduction division. In this way, this is the basic definition or meaning of the cell division. So how the cell division works, how the cell signaling is given by the cell to the nervous system, or one cell to another cell, or inside the cell its activities, how the signaling is be there. So we will see one by one. First of all, we will see a cell cycle. What is cell cycle? The period from the end of one division of a cell to the end of the next division is called as cell cycle. Means one cell which requires its division and forming the two different cells, this period is totally called as a cell cycle. Once again, I will define you the period from one end of the cell division to the end of the next division is called as cell cycle. Normally, these dividing eukaryotic cells mostly have a cycle of 10 to 30 hours. Means one cycle is completed near about within 10 to 13 hours. It is depending on the type of cells and type of organism. Of this period, only about 30 minutes to 1 hour is taken by actual division phase, means last phase we can say. Thus, the cell cycle is divisible into two main phases, that is growth phase or interphase, the growth phase or interphase, and division phase. Means in first half phase only the growth is there, and in the next half phase the process of cell dividing is actually started. Means the cells divide into two cells. A growing cell undergoes a cell cycle. It is a recurrent series of events or changes each dividing cell possesses through a cycle. It has a mechanism. Before cell divides, it must double its mass and duplicate all its contents. Then and then only the cell can divide, because it requires to have a minimum requirement of the contents, and therefore the material doubles, and then whenever it becomes double, half is divided into another, and therefore two cells are formed. After division, the duplicated material passes into the two daughter cells. This orderly sequence of events is termed as cell cycle. By another way, we can say that this orderly sequence of events is termed as cell cycle, means interphase and division phase. So it includes two different subphases again, means total 4 phases we will study here, and the cell cycle is completed therein. So for many years, cytologists, means the scientists, those who are working on the cell is called as cytologist, where cytology means study of cell, were concerned mainly with the period of division, that is mitosis, in which dramatic changes like prophase, metaphase, anaphase and telophase are visible under light microscopes. And the another phase, that is interphase, was considered as a resting phase of the cells, means the cell for pause its activities for some periods, for some time, or we can say it take a rest, therefore it is a resting phase. However, cell spends most of their lifetime in interphase, but actually during this phase, cell itself prepare before duplication or division of the cell, means the next division is started in the period, we can call it as a preparatory phase, that is mitosis or meiosis. Some differentiated cell types generally divided, rarely, and in mammals, nerve cells do not divide at all after birth. The nerve cells do not divide at all after birth. Thus, for a human nervous, the interphase period lasts the entire lifetime of a person. The interphase period lasts the entire lifetime of a person. The period of cell cycle depends upon the cells. The period of cell cycle depends upon the cells and the external factors also, which are continuously varies in species to species, or different species show different variations in the factors, because every animal is living in different environment, therefore the external factors are variable. In general, it could be 24 hours or less in different species. Thus, cell cycle may also be defined as those changes which occur during cell growth and cell division. So we will study these two stages, that is interphase and growing phase, we can call it growing phase, and dividing phase, or interphase and dividing phase, the actual division phase. So these two pages we can divide them again into two different pages, that is 4 pages for our convenience. So these are the stages of cell cycle: G1 phase, S phase, G2 phase and M phase. So these are the 4 phases of the cell cycle, that is growth phase and maturation phase, or mitosis phase or meiosis phase, whatever the type of the cell may be. So these are the growing phases and these are the dividing phases. So we will study one by one. Cell cycle includes the period from the beginning of one cell division to the beginning of next cell division. The time taken to complete one cell cycle, is called as generation time. This is generally called as generation time. And the cell cycle involves two main phases, that is interphase and mitotic phase, or we can call it a dividing phase, or interphase or growth phase, and dividing phase or mitotic phase. These are the two main phases, and in these two pages we can study these 4 different pages. How we will see one by one. This is the picture of diagrammatic presentation of the cell cycle: G1 phase, S phase, G2 phase and M phase. So G1 is the growth phase one growth phase 2. In between these two, one S phase or synthesis phase, the material is synthesized for the development of that particular activity. And finally, mitotic phase or dividing phase or M phase. So these are the four phases: G1, S, G2, M. So in G1 phase, the initial growth or pre-DNA synthesis is occur, or we can say the whatever the material is collected there, that material is dividing, that material is growing fastly, and this material which is required for the production of DNA is prepared in this phase, and this phase is generally of 10 hours out of 30 hours. We have seen 10 to 30 hours, so 10 hours maximum period is given to the G1 phase. Then in second S phase, this pre-DNA material is converted into DNA material, and that DNA material is synthesized. Therefore, this phase is called as S phase or synthesis phase, and this occurs, or this phase takes about 8 hours. Then second is G2 phase. After synthesis, it grows in long manner. Like this is pre-DNA phase, this is post-DNA phase, and this is DNA phase. We can say that this is the preparatory material and this is developing material. So DNA material is synthesized, or second growth phase is started. Whatever the material is developed here, it starts to grow in its size, number, whatever it may be. And this period or this cycle time takes about 5 hours. This one is 10 hours, 8 hours and 5 hours, and only last phase takes 1 hour, in which the cell divides into 2 cells. But this within this 1 hour, it goes through mitosis. We can say generally body cells we study in cell division. So generally this phase, division phase, takes 1 hour, and during that 1 hour it passes through 4 stages, generally called as first stage prophase, second metaphase, third anaphase, and fourth telophase. Means even in these 4 phases, whatever the DNA are synthesized here, whatever the DNA are grown here, they all are come in the single line in prophase. Nay, we can say they prepare their couples, they prepare their pairings, chromosome pairings. Then they come on the equator line in metaphase. Then they start to stretches to the both ends in anaphase by forming ester, and in telophase the pinching between these cells, which is a clear line, demarcating line, that the division of cell actually is started, and 2 cells are separated in telophase, and in this way this cycle is completed here, and 2 daughter cells are formed from 1 cell. So in this way this cell cycle completes, and 1 cell is divided into 2 cells. So this is the complete picture, complete diagrammatic presentation of the cell cycle. We will see the details of the cell cycles. What happens actually there too. Actually I have explained in brief, but we will see in details. Interphase. It is the preparatory phase, where the preparation of material for the development of the for the division of the cells is prepared between 2 successive cell divisions. It is the non-dividing stage of the cell, means only preparation is being here, and its nucleus is called as interphase nucleus. Whenever the cell undergoes cell division, at the beginning the nucleus is called as interphase nucleus, because it is also prepared to send the information to the materials that we are ready, we must keep ourselves ready to divide the cell. So it is called as interphase nucleus. It was considered as a resting phase of the cell. However, the cells spend most of their lifetime in this period or in this phase, but actually during interphase the cell itself prepare before duplication or division of the cell. Means this cell is only the preparatory phase. This one is the preparatory phase. Further, it is, or this is divided into three sub-phases, that is G1 phase, growth number 1 phase, S phase, synthesis phase, and G2 phase, growth number 2 phase. So these are the basic types of these interphase. So we will see one by one. G1 phase, it is also called as post-mitotic gap phase, or first gap between the end of mitosis and the start of DNA synthesis. Means pre-DNA synthesis phase, or we can say synthesis, is started actually in this phase. The time span required for this phase varies greatly in different types of cells in same organisms. Even though the organisms are same, but division time may be different in different organisms. And as we have seen in previous figure, this time is about 10 hours in first phase. During this phase, the cell synthesizes enzymes necessary for DNA replication. We have seen in figure how the DNA synthesis, DNA pre-synthesis and DNA post-synthesis is observed. So there is arrested stage of cell cycle, that is G0 phase. It is arrested at specific point of G1 phase. Therefore, we generally call it as arresting phase. Anytime, anywhere, in G1 period, it may take a pause or stop. In this condition, cell may be considered to be withdrawn from cell cycle. So this may be happen sometimes, not every time. Accidentally it happens. It may last for days, months, or even years. Example, nerve cells that do not divide. Therefore, this phase is most variable phase of the cell cycle. Given phase is the most variable phase of the cell cycle. And in this phase, the regulation of cell cycle occurs, during which it decides whether the cell will start a new cell cycle or become arrested in G0 state. And once the G1 checkpoint has been passed, the cell goes on to complete a new cycle, and this phase takes maximum time of about 9 to 10 hours. So this is about the phases of the cell cycle. Second phase we have studied already is the S phase or synthesis phase. It is characterized by the synthesis of DNA, because actual synthesis is here in S phase, and also synthesis of histone proteins is occur in second stage S phase. Consequently, DNA content of cell becomes double, as was demonstrated by pulse change experiment, because the pulse change experiment shows that the DNA content becomes double and ready to divide in two different cells. Each chromosome reveals or shows two chromatids, and once the cell enters S phase or synthesis phase, the cell has committed itself to divide. Means it is ready for the division, and this phase takes maximum of about 8 hours. So first stage about 9 to 10 hours, second stage about 8 hours, and third stage G2 phase will take 5 hours. We have already seen in diagram. So growth second phase or post DNA synthesis phase is pre-mitotic or second gap phase, because after this phase the cell is ready for dividing two cells, and gap phase between end of DNA synthesis and the start of mitotic phase or M phase. It is characterized by intensive cellular synthesis of various enzymes, ATPs, and the factors which are necessary for chromosome condensation. Means in G2 phase, not only the post synthesis occurs, but DNA cells make themselves very prompt, particular, and ready for the further activity. Therefore, they synthesize not only DNA material but enzymes, then activate adenosine triphosphate molecules, and condensation of the chromosome also seen in this G2 phase. During this phase, the cell contains double the amount of DNA. As we have seen in previous phase, it becomes double. So here the amount of DNA doubles, means here double and here again double, means 2 and 2 4. Therefore, it will make a maximum amount and ready for division of the cell in the next stage, because next stage is division, mitotic phase, as compared to the original diploid cell, 2 cell, means 2C becomes 4C. It doubles and it lasts for about 5 hours. So this is about the G1 phase, S phase and G2 phase. In next phase the cell division will start, but it will go through the 4 stages: prophase, metaphase, anaphase and telophase. So we will see these phases in the next period. Till then we will see you. Okay.